Hello everyone, good evening. So our today's topic is open chain structure of glucose. Yesterday we have done that we have proved, or we have there is a CHO group, there is wait. Yes. So open chain structure of glucose. So in this case, what we have done, we have proved that there are 6 carbons from its molecular formula. Then there is a straight chain with the reaction with Hi and red phosphorus. It has an aldehyde group because it reacts with HCN and NH2OH, that is hydroxylamine. Okay, it has a 5 OH groups. I am good now. Vasant, thank you. 5 hydroxyl group because it forms glucose pentaacetate with acetic anhydride, and there is one primary alcoholic group because when it reacts with the HNO3 it gives saccharic acid. Okay. Now let us discuss its open chain structure. So from all these reactions it is clear that it has glucose has one aldehyde group, that is CHO group, one primary alcoholic group, that is CH2OH. Now since in all glucose has 5 OH groups, one of which is CH2OH, so other 4 OH group must be secondary. So that means there are 4 secondary alcoholic groups, that is CHOH. Okay, because these are in between CHO and CH2OH. Okay, so there are 4 CHOH groups. So open chain structure is considered to be CHO, then CHOH, again CHOH, 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 and at the end CH2OH, right? So in the molecule of glucose it has, see, this is chiral carbon, this one is also chiral carbon, again chiral carbon, chiral carbon. Okay, so there are 4 chiral carbons. So how many stereoisomers are there? You know, number of stereoisomers is equal to two raised to power n, where n is number of chiral carbons. So n is 4, that implies 2 raised to power 4. So total, there are total 16 stereoisomeric forms. Okay. So there are 16 stereoisomeric forms. So total groups are, pair of enantiomers are 8. There are 8 pairs of enantiomers. Okay. Now the exact special arrangement of different OH groups, it was determined by Emil Fischer. That exact arrangement that was given by Emil Fischer, a German scientist. Okay, and that configuration is like see CHO on the first position, means most in Fisher projection formulas, most oxidized carbon is at the top. So you know CHO is most oxidized and CH2OH is least oxidized. Okay, so that's why we write it like CHO, then one, two, three, 4 and then CH2OH. Okay, so on the second carbon there is HOH, on the third carbon it is OH, H, on the fourth carbon it is HOH, HOH. So this is the exact arrangement, spatial arrangement of H and OH. Okay, so this is called glucose, and you know in gluconic acid obviously CHO changes into, yes, CHO changes into COOH and rest part remains same. HOH, OH, H, HOH, HOH. We should keep in mind all these arrangements. Only see at third carbon it is different. Rest is HOH. Okay. 1, 2, 3, 4, 5, 6. Sorry. So this is gluconic acid. In saccharic acid, there is COH group on both side and rest is HOH, OH, H, H, OH, H, OH. 1, 2, 3, 4, 5, 6. Okay. So OH group at C3 chiral carbon is oriented towards left, while the remaining 3 OH groups at C2, C4, C5, these chiral carbons, they are oriented at right. See, OH group at C2, C4 and C5, C2, C4, C5, C2, C4, C5, they are oriented towards right, but in the third at C3 carbon, this OH is oriented to the left. Only this is the difference. Okay, got it? Any confusion in the structure? Yes beta, कर लोगे बेटा? स्ट्रक्चर ड्रॉ कर लोगे? Now let us talk about the configuration. So correct name of glucose is actually, see, correct name of glucose is D plus glucose. Correct name is D plus glucose. You know now glucose is optically active, so obviously its enantiomer should be L negative. Its mirror image should be L negative glucose. If it is D plus glucose, then its mirror image should be L negative glucose. So keep in mind this letter capital D and capital N used before the name of glucose represent the configuration of the OH group at the penultimate carbon atom of the carbon chain. Okay, penultimate means second last carbon. See, when we write it in the vertical arrangement, then second last carbon, this is the second last carbon. In these second last carbons, OH is on the right side. So, if OH is on the right side, then yes, if OH is on the right side, then it is called D. No, dextro and levo is different. This plus and minus sign means plus is dextro, plus means wait, plus means dextro rotatory and negative means levorotatory. Okay. So, but keep in mind this D and L, they have no relation with the sign of optical rotation. So, say if a glucose or maybe any carbohydrate has D configuration, so it may be D plus or D negative, means configuration is D. So, it may be either dextrorotatory or levorotatory and vice versa, like L may be plus or L may be negative. This sign has no relation with D and L. Okay. So, D glucose is dextrorotatory. Best example is glucose and fructose. See, D glucose and D fructose. Let us write it like so. D glucose is dextrorotatory, but D fructose is levorotatory. That means it is D plus glucose, but this is D negative fructose. Plus means dextrorotatory, minus means levorotatory. Okay, and D, L and are simple configurations. So, to assign D and L configurations to monosaccharides, जैसे सिंपलेस्ट कार्बोहाइड्रेट कोई भी लो, जैसे ग्लिसरल्डिहाइड ले लिया हमने, तो देखो in glyceraldehyde there is CHO and here it is CH2OH. Okay. CHO, CH2OH, here it is H and OH. So, this is chiral carbon. This carbon is chiral. So, this is actually D plus glyceraldehyde. Okay. And its mirror image is L negative glyceraldehyde. Okay. Because it contains one chiral carbon, as it exists in two enantiomeric forms. So, all the compounds which can be chemically correlated to plus isomer of glyceraldehyde are assigned D configuration, or all those compounds which can be correlated to minus isomer of glyceraldehyde are assigned L configuration. Okay. So, if a monosaccharide is assigned D configuration, that means OH group on the last chiral carbon, means obviously second last carbon is last chiral carbon, it lies towards right hand side. Am I right? For example, best example is posed by glucose, even fructose. See, 1, 2, 3, 4. CH2OH, CHO. See, HOH, OH, H, HOH. It has no relation with D and L, but second last carbon or last chiral carbon. See, in last chiral carbon, OH is on the right side. That's why it is called D. Okay, it is dextrorotatory, that is experimentally proved. Nothing to do with D and L. Okay, so this is D glucose. Okay, even if we draw structure for ribose, there are 5 carbons, HOH, HOH. Now last one, it is also HOH. So see, it is on right side, so it is again ribose. It is levorotatory, that's why it is called D negative ribose. D negative ribose. Okay. Now, if I write this glucose as, sorry, just see, CHO means its mirror image, 1, 2, 3, 4. Mirror image of D glucose, OHH, HOH, HOH, sorry, OHH, HOH, OHH. Now last one, OH is on the left side, so it is L. It is L. Okay. I think D and L configuration, it is clear to you. Understood? Now next part, that is cyclic structure of D glucose. So to assign cyclic structure, first let us discuss the limitation of open chain structure. So limitations of open chain structures are important. Okay, limitation of the open chain structure. So see, first limitation, you know, D plus glucose react with HCN and D plus glucose that react with HCN and NH2OH, and on that basis we said that it has CHO group. But if it has free CHO group, obviously D plus glucose should give all the tests of aldehyde, all the characteristic test of aldehyde, but D plus glucose does not give or does not undergo certain characteristics test of or reactions of aldehydes. Okay. So, there are many reactions which does not undergo. Okay. Otherwise, if D plus glucose has a free CHO group, then it should give all the tests. Like glucose does not form CN sodium hydrogen sulfide addition product. You know, that is the characteristic test of all aldehyde compounds. Okay. It does not react with NaHSO3. Even aldehyde ammonia duct is not formed, means it does not react with aldehyde, does not react with glucose does not react with 2,4-DNP derivative. You know that is the test for carbonyl compounds. Even it does not respond to Skipp's reagent test. So, this limitation proves that D plus glucose does not have free CHO group, because it does not form sodium bisulfide addition product, it does not react with 2,4-DNP derivative, it does not respond to Skipp's reagent test. These are the characteristics test of aldehyde group. Now, say glucose reacts with NH2OH, that we know it forms oxime, but glucose pentaacetate does not react. So, second limitation is posed by that glucose reacts with NH2OH to form an oxime, but glucose pentaacetate, when we form glucose pentaacetate, we have shown that there is a free CHO group, okay, but glucose pentaacetate does not react with NH2OH. It implies that aldehyde group is absent in glucose molecule, glucose pentaacetate, sorry. In glucose pentaacetate, there is no CHO group. Even D plus glucose it exists in two isomeric forms. That is alpha glucose and beta glucose. Okay. Now, alpha D plus glucose and beta D plus glucose. So, that is not explained by open chain structure, right. So, these are the three limitations of open chain structure. Even other limitations are also there, but we have to cover these three limitations. On the basis of these three limitations, we have to draw another structure of glucose. The open chain structure is not the actual structure of glucose, because if it has open chain structure, then CHO group is totally free. So, it will give all the characteristic test of aldehyde group, which is not given by the glucose. And glucose pentaacetate, we have shown in the structure that CHO group is free, but it does not react with hydroxylamine. So, that means in glucose pentaacetate there is no free CHO group, there is means CHO group is absent. glucose exists in two isomeric forms, that is alpha and beta, that is also not explained by open chain structure. Okay. So now let us discuss the cyclic structure of glucose. So in cyclic structure of monosaccharides, there are two forms actually, hemiacetal forms. First is like furanose. This is furan, 5 membered, okay. And other is pyran. Okay, furan and pyran. Now see, in monosaccharides they contain number of hydroxyl groups, aldehyde group or keto group. Therefore, any one of the OH group, usually C4 or C5 in aldohexoses and C5 or C6 in ketohexoses, they may combine with the aldehyde or keto group to form the intramolecular hemiacetal or hemiketal. That is why monosaccharides have 5 or 6 membered rings. They have 5 or 6 membered rings. Okay. So 5 membered ring containing one oxygen atom, because of its similarity with furan, they are called furanose form. One is furanose form, and 6 membered rings containing one oxygen atom, because of its resemblance with pyran, they are called pyranose form. See, this is furan, 5 membered ring with one oxygen atom. Oh sorry, this is O, okay. 6 membered ring with one oxygen, so that is called pyranose form. This is 5 membered ring, 6 membered ring, okay. Because of their similarity with furan and pyran, they are called furanose form and pyranose form, okay. So let us discuss the cyclic structure of glucose now. See, you know, glucose has CHO, then HOH, at third carbon there is OH, H, OH, 1, 2, 3, yes, 4, and then CH2, OH. This is the open chain structure of glucose. In open chain structure, you can say there is free CHO group. This is open chain form, okay. Now 1, 2, 3, 4, 5 and 6. Lone pair of oxygen OH attached to carbon, a fifth carbon, that attack here, okay, and double bond, this pi bond shift towards oxygen. So when this lone pair is given to this carbon, this oxygen gets positive charge, and because of that this H as a positive ion removed from here and attached to oxygen, this oxygen which is negatively charged, okay, because of the transfer of pi electrons, and we get HOH even on the first carbon. So when this HOH is formed, this HOH may be orientation like OH on the right side or OH on the left side. So see 1, 2, 3, 4, 5 and 6. Sixth carbon is like CH2OH. So there is one oxygen here. Okay. H, OH, OH, H, H, OH, H, OH. If it is like this on the first carbon, okay, if OH is on the right side, this is called alpha D plus glucose, or it is also called alpha D plus glucopyranose, alpha D plus glucopyranose, because see 1, 2, 3, 4, 5 and oxygen is sixth. So obviously 6-membered ring is there. So pyranose form is formed, or on the first carbon OH may be on the left side. If it is on the left side, this is called beta D glucose. Sorry, on the third carbon there is OH, H. Just look at this configuration. Rest configuration remains as such. Only at the first carbon it happens. Okay. H, OH, H and OH. Right? First, second, third, fourth, fifth, sixth. Okay. This is called beta D plus glucose or beta D plus glucopyranose. Okay. Say in alpha D plus glucose, OH on the first carbon is at the OH at C1 is towards right, and in beta D glucose OH at C1 is at the left, towards left. Okay. So this alpha and beta, say alpha D plus glucose and beta D plus glucose, they differ in configuration only around C1. So such a pair of stereoisomers are called anomers, and this C1 carbon is called anomeric carbon. So I will write it alpha and beta. Okay. So such a pair of anion stereoisomers, we can say, which differ in configuration at C1. Are called anomers. That means alpha and beta D plus glucose, they are anomers, and the C1 carbon is called anomeric carbon. Called anomeric carbon, or sometimes we also call as glycosidic carbon. It is also called glycosidic carbon. Okay. Because this COC linkage is also called glycosidic linkage, right? Okay, so this is about the cyclic structure of glucose. Now say, as we have done it, 1 2 3 4 5, okay, 6. First HOH, second HOH, third OHH, HOH, CH2OH. So this is alpha D plus glucose, right? So it, to write it like this, this is Haworth projection formula, okay. This one is Fisher projection formula, okay. Let us write it like here only. Okay, so now these are called, this vertical line, that is called Haworth projection formula, okay. Sorry, Fisher projection formula, okay. Now this is Fisher projection formula. Now let us write Haworth projection formula. Now to write Haworth projection formula, it is like this. The one on the left side, that is at the top, and right side, that is at the bottom. Now this is 1 2 3 4 and 5. 1 2 3 4 5, and this is 1 is 6th, okay. So in this formula, just write say on the first carbon H on the left side. So H above the plane, OH below the plane, okay. Then again HOH, HOH, then OH, H, HOH, and terminal carbon, that is terminal CH2OH group, is always above the plane of the hexagonal ring, that is CH2OH and H. So this is the Haworth structure. And this one is Fisher projection, okay. That is for alpha D plus glucose. This is called alpha D plus glucopyranose, okay. Now beta, for beta there is only one change, because on the first carbon there is a change, 1 2 3 4 5 6, OHH, HOH, OHH, HOH, and then like this. This is beta D plus. So let us write its so it is OH, H, H, OH, OH, H, C, first, second, third, fourth, fifth, sixth. First, second, third, fourth and fifth. हाँ बेटा. So this is again H, OH, CH2OH H. That's it. This is a Worth projection formula, alpha D plus glucopyranose. Okay. Now structure of fructose. Structure of fructose. So fructose is actually the name. This fructose, is the most important of all the ketoses. It occurs free along with glucose in the form of invert sugar, So in honey, sweet fruits, and hence the name is fruit sugar. Fructose is called fruit sugar. Okay. So structure of fructose. See, in fructose there is one CHO group, CO group. It is ketosis. Like first is CH2OH, CO, third, fourth, fifth structures are same, means configuration is same as that of glucose. See, there is on the third carbon there is OHH, HOH, HOH, right? So now in this case, how do we draw the structure? Obviously 2, 3, 4, 5 and 6. So in this case, again this carbon on the fifth lone pair of the fifth position OH group, that attack here. Again same situation. So see 1, 2, 3, 4 and 5. 5-membered ring is formed. It is like, say 1, 2, 3 and 4. CH2OH. So this first CH2OH group and then OH. OH is either on the right side or left side. See, if it is on the left side, obviously CH2OH on the right side. And at third carbon, it is first, second. At third carbon, there is OHH. On the fourth carbon, there is HOH. Now, there is OH on the right side. HHOH. Okay. So now this is called alpha D negative fructose, because on the second carbon, see, on the second carbon OH is on the right side. Here OH is on the left side. So, it is also called alpha D negative, yes, fructofuranose, because it has a resemblance with furanose. This is called beta D negative fructofuranose. Okay. So, we can draw its structure. So, this is open chain form, and these are closed one. Okay. Now, to draw their Haworth structures, see, there is 1, 2, 3, 4 and 5 membered rings. Okay, beta. Sorry. See, on the second carbon there is a CO group, and on the fifth carbon this oxygen that get attached to second carbon. So, see, 1, 2, 3, 4 and oxygen. So, 5 membered ring is formed. On the second carbon the OH is formed. So, OH is either on the right side or left side. So, if OH is on the right side, this is called alpha, and if OH is on the left side, that is called beta. Okay, rest remains same. They are just like glucose. On the third carbon OHH, fourth carbon HOH, fifth carbon HOH. Okay. So, to remember this whole thing, now see, on the first carbon means second carbon, if OH is on the right side, so obviously CH2OH is on the left side. This is first carbon second. On third carbon, keep in mind it is always OHH, HOH, CH2OH. So, let us draw its Haworth structure. It is like this. So, this is second carbon, third, fourth and fifth. Okay. So, the group on the left side is above the plane, CH2OH, OH, and then third OH, H, fourth carbon H, OH, and on fifth carbon it is CH2OH and H, because on last carbon is always at the top. Done. This is alpha D negative fructofuranose. Okay. Now, beta. For the beta, OH is on the left side. So, देखो, see 1, 2, 3, 4 and fifth is oxygen. And there is CH2OH. So, if OH is on the left side, so obviously CH2OH on the right side. 1, 2. Third carbon, you know, it is OHH. So let us write. I think you can write it easily now. Okay. So on the second carbon, third, fourth, fifth, second carbon OH on the left side. So top, CH2OH on the right. Then OH, H, fourth H, OH, and last carbon is always at the top, CH2OH and H. So this is called beta D negative fructofuranose. And keep in mind, all these monosaccharides, whether aldoses or ketoses, in their hemiacetal form and hemiketal forms, they behave as reducing sugars. Okay, they behave as reducing sugars. So अब आपको मुझे लगता है कि दिमाग में आना चाहिए कि fructose has keto group. Fructose has keto group, and you know, ketones do not reduce tolerance reagent or failing solution. So how will this happen in fructose? Because fructose has keto group, but it is a reducing sugar. So keep in mind, you know, they are always in equilibrium actually. Only this part plays the role. You know, this whole part remains same in glucose, mannose, fructose. This whole part remains same. Only first and second carbon, they are involved. Okay. So first of all, what happened, you know, this is CHO group and this has H, OH. So this is involved in enolization. The CHO is involved in enolization. 1, 2 enolization takes place. This is, you know, D plus glucose. D plus glucose is reducing sugar because it has CHO group. Okay, in hemiacetal form. How to name these compounds? Okay, D, I have already told you that D means at the second last carbon OH is on the right side, and this plus minus sign, that is experimentally proved. Okay, now see, this H is shifting to O, means enol form is formed, CHOH double bond, COH, the CHOH thrice and CH2OH remains as such. Okay, now look at it. When this enolization is converted into means this enol form is converted into keto form. This is, you know, enol form, है ना? En diol. This is en and 2OH, है ना? En diol. So say this CHO group. Now the CHO group remains as such. Now this HOH, it may be OHH. Okay, so this is called D plus mannose. So D plus glucose as well as D plus mannose, both are reducing sugars, that is clear, but this is in equilibrium with CH2OH, C double bond O, CHOH, Beta Saad, I have been in this field since 2003. So you can imagine, इतनी बार पढ़ोगे तो किसी को भी याद हो गया. D negative fructose. So you will start with glucose or mannose or fructose, all of them are in equilibrium. Always this same equilibrium mixture is obtained, okay, either with glucose, fructose or mannose, and that's why fructose reduces tolerance reagent and failing solution, although it does not contain CHO group, okay. Fructose has no CHO group, but because of this equilibrium mixture they can form. No, beta, racemic mixture is something different. Racemic means if there are mirror images, you know, glucose and mannose, they are not mirror images, they are apimers. Yes, new term. See, if configuration differ at C1 in aldoses and C2 in ketoses, they are called anomers. If configuration differ at any other carbon, then that they are called apimers. So D glucose and D mannose, D plus glucose and D plus mannose are apimers. Racemic, in a racemic mixture, in an enantiomer, that is pair of enantiomers, they have mirror image relations. They are actually diastereomers. Okay, those isomers which have no mirror image relation, okay, they are called diastereomers. So they are actually diastereomers, okay, and D plus glucose and D plus mannose are called apimers because they differ at configuration at around C2 only. So that's all for the day. I think you can do it easily. One more thing I would like to tell you, because that is important for your competitive exam. Last one. So in carbohydrates we will do this part only, that is in formation of OSA zones. In competitive exam, question asked was, and it will be in this year, that in the formation of OSA zone, how many phenyl hydrogen molecules are used? How many phenyl hydrazine molecules are used? That question may be there in this year. See, you know, we have structure like this. So first of all, phenyl hydrazine. So when phenyl hydrazine react, you know, O of CHO and H2 of phenyl hydrazine, that will be removed and we get this molecule. Okay, phenyl hydrazine is in excess. Okay, so second molecule of phenyl hydrazine comes into picture. The CH5NH2 and NH3, they are removed. Okay. OSA zone, beta, this whole thing I let you know. When second molecule is involved in oxidation of alcoholic secondary alcohol into ketone, and then we get next molecule, water is removed. Sorry. So this is called glucosazone. This is actually osazone. Okay. So in the above reaction, during this osazone formation, 3 moles are used. Okay. 3 moles of phenylhydrazine are used. Okay. First mole reacts in the usual manner, like the aldehyde. Second molecule जो है, that second mole oxidizes secondary alcoholic group at C2, and third mole that react with keto group to form the osazone. So this is called osazone actually, beta. This is called osazone. So in osazone formation, say C1 and C2 are involved. Only C1 and C2 are involved. That's why all monosaccharides which differ in configuration at C1 and C2 give the same osazone. That means glucose, mannose and fructose, all of them will give same osazone. They will give all same osazone and involved on 3 moles of phenylhydrazine. Okay, beta. So that's all for the day. Thank you so much. Bye bye to all. If you have any doubt, please do ask. Yes, beta. Anyone who has a doubt? Yes, beta. Any doubt? Okay, bye bye then.